Hello, everyone. Welcome to another gripping talk. Today it's my pleasure to introduce our speaker, Dr. Francesca Flodini. Francesca completed her PhD in the lab of Professor Isabel Roditi at the University of Bern, where she worked on transcriptional regulation in African trypanosomes. She then moved to the US and is currently a postdoc in the lab of Professor Kurt Deitch at Weill Cornell Medicine. Her work now focuses on on malaria parasites. You know what they say: new job, new parasite. And she's particularly working on looking at antigenic variation of parasite to evade the host immune system, and she's doing this at single cell level. So we are very excited to have her here. Please join me in welcoming Francesca.
Thank you. I'm also very excited to be here and tell you a little bit about what I did during my postdoc in Kurt Deitch lab, where I mainly worked on the mechanism of immune evasion of the malaria parasite Plasmodium falciparum. And please feel free to interrupt me at any point if you have any question or something that is not clear. I feel like I have a bit of an introduction that is probably unnecessary for this crowd, but I'm going to go quickly through that anyway. So malaria is a disease that is caused by protozoan parasite of the genus Plasmodium. And as you probably know, there are 5 species of Plasmodium that infect humans. But by far, Plasmodium falciparum is responsible for the majority of these numbers, so the majority of the malaria morbidity and mortality. And they are transmitted between humans by the bite of an Anopheles mosquito. And here you can see the distribution of malaria throughout the world, and you can clearly see that is widespreading throughout the tropical and subtropical region of the world. But that being said, the majority of the cases are in Africa, which is where Plasmodium falciparum is mainly endemic. So just a quick overview of the Plasmodium falciparum life cycle, even though, again, I think you are aware. After a bite of an Anopheles mosquito, the sporozoites make their way to the liver, where they go through a first round of replication. And then merozoites get released into the bloodstream. And there they invade the red blood cells. And they start what is called the asexual cycle of replication within the red blood cells, where they start to replicate within a red blood cell, they release new merozoites, and then this merozoite can invade a new red blood cell. And then eventually some of these parasites are going to differentiate to gametocyte, which is the transmissible form, so the form that can continue the transmission in a new mosquito. But the part that I'm particularly interested in that I worked on in the lab is this, let's say, approximately 48 hours asexual cycle. So I want to give you a bit more details about it. So when Plasmodium falciparum infects a red blood cell, it modifies this cell in a way that makes it less deformable. And as a consequence, this cell cannot really pass through the spleen as efficiently, as the spleen is the main organ for the quality control of our red blood cells. So to avoid that, what the parasite do is that they can export a protein on the surface that is called PFMP1. And this protein allows the cell to cytoadhere to the vascular endothelium and as a consequence, avoid passing through the spleen. Now, this is also one of the main reasons of malaria pathogenicity, in the sense that depending on where these red blood cells cytoadhere, they can cause a different clinical manifestation. They can, for example, cause cerebral malaria if they adhere in the brain, or pregnancy-associated malaria if they adhere in the placenta. But looking at this from the point of view of the parasite, now, if before the parasite was completely hidden within a red blood cell and our immune system could not recognize that there was a parasite in it, now they expose themselves, so our immune system can build antibodies against this PFMP1, and the infection can be cleared through antibody clearance. So to avoid that, the parasite can actually exchange the isoform of PFMP1 that they export on the surface in this process called antigenic variation. And as you know, because of people that work here also, antigenic variation is not unique to Plasmodium. It's actually a mechanism that several microorganisms use to escape immune recognition. So it's common to parasites, but also to virus and bacteria. And it's normally connected to a family of genes that encodes for different isoforms of the antigen exposed on the surface. And in the case of Plasmodium, these are called VAR genes. There are approximately 60 or so VAR genes per Plasmodium isolate. They are very different between different strains of Plasmodium, but they are always, as you can see here, these are the 14 chromosomes of Plasmodium, and they are always in clusters, either in the subtelomeric regions or some internal clusters. And the dogma in the field, which in the end of my talk, hopefully you're not going to believe as much anymore. The dogma in the field is that their expression and silencing is mutually exclusive, which means that basically it is thought that in any given parasite at any given time, there would be only one VAR gene on and all the others are off. But then the parasites are able to switch to a different one, so the first one will be silenced and then a second VAR gene will turn on, which is thought to cause these typical waves of parasitemia, typical of a malaria infection, that are thought to be fairly homogeneous wave with the same PFMP1 on the surface. So there are a series of questions that are still open about this process, and there were two things that I was particularly curious when I joined Curve Lab. One of them is how is switching coordinated? So like if we envision this population of parasites in an individual, there are millions of parasites at the same time. How do they coordinate which PFMP1 is on? Because if you imagine you have millions of parasites in your body and each of them decides to express a random VAR gene, then obviously and there are only 60, the repertoire will be exhausted in the first wave or maximum 2. But if this is not the case, so that my question was, how do parasites decide which VAR gene to turn on and like sort of communicate that with each other? And the second question is sort of connected to that, is like even if they coordinate their VAR switching, there are only 60 or so VAR genes. So what happens once they exhaust this repertoire? Because there are instances of chronic malaria infection that can last in extreme cases even up to 10, 13 years. So eventually these 60 or so VAR genes are going to be exhausted anyway. So my question was, what happens then, like once our antibodies have taken control of the different isoform of PFMP1? And in trying to answer some of these questions, I try to take some ideas from other systems that do mutually exclusive expression. One of them is olfactory receptor genes. So you might know that our olfaction relies on the fact that there is a family of genes. There are around 400 in humans and more than 1,000 in mice that are mutually exclusive expressed in each of our olfactory neurons. So once our neurons develop, only one of these genes is on and all the others are off. And I thought there was a very interesting paper that came out around 10 years ago, where they explored this at a single cell level. And just to explain what you're seeing here, so each of these columns is an individual neuron, and each of the color in the column is a different olfactory receptor gene. So what you can see here, they basically isolated olfactory neurons at different steps of their maturation, and you can clearly see that the expression goes up the more mature they get. But also that what I found really interesting was that when they looked at like relatively early neuron, they could see that they were basically expressing several genes at the same time, but at a very low level. And something similar happens also in another organism that you might know, which is Trypanosoma brucei. And as you probably are familiar, trypanosomes like shuttle between a mammalian host and a tsetse fly host. And when they are in the mammalian host, they face a similar problem as Plasmodium. So they are swimming freely in our bloodstream, so they need to go through antigenic variation as well. And they do that because they have a family of genes as well called BSG. And even though they are a bit hard to study in the bloodstream form because there are a lot of them, there was a group that actually studied them at the metacyclic form, which is the last bit of the development within the tsetse fly. So they did single cell RNA-seq at this step of the development of the trypanosome, and what they observed was something very similar to what was shown in olfactory receptors. So that before committing to expression of an individual BSG, they had the low expression of several BSG, but again at a very low level. So when I saw these two different stories, I was curious to if something similar could be happening also in Plasmodium. And before I go through the results, I just want to quickly explain you how we look at VAR genes in Plasmodium. So we have a set of primers that recognize different VAR genes in the strain that we use in the laboratory. And we can do a qPCR, and we will display either as a pie chart, and each of the slices in the pie is a different VAR gene, or as a bar graph where every column is a different VAR gene. And you can see that this is again the population level, but you can see that more or less this population is mutually exclusive, expressing this yellow VAR gene. So, but when I started looking at this a bit more systematically, I started noticing that wasn't always necessarily the case. So here is like a clonal population, and you can see again that there, in this case, there is a major VAR gene on. But then I started observing cloning that look like this. Like basically they didn't have a main VAR gene expressed at all. And when I stumbled through this, I was, we need to look at this in a more systematic way. So we did a very fun experiment where we took an initial parasite population, we put it through a cloning plate. So we were putting individual parasites in individual wells. And then we will isolate the new clonal population as then extract RNA and check what their VAR profile was. And every time we were getting a new group of clones, Kirk was like, "Oh my God, this is so exciting! Let's do another plate." So we did this in a couple of generations, and this is like some of the results that we got. So you can clearly see that populations were very heterogeneous at the point of view of which VAR genes they were expressed. So you can see that even when you start from a population that is expressing one VAR gene, its daughter clones would be really different. Some of them were expressing the same VAR gene, but some others were in this low many state. And population of parasites seem to be able to go from one state to another fairly frequently within like generation of clones. But obviously, there's something missing when you look at this, which is what is the contribution of individual cells to this population. So if you look at a population that looks like this, is it that actually individual parasites look like this, or it's more that the population is very heterogeneous from the point of view of which VAR gene they are expressed? So that's when I started thinking about we really need to look at this process at a single cell level, because again, even from the dogma that I was telling you before, that expression is mutually exclusive, everyone has looked at it at a population-based, with population-based assay, so RNA-seq or qPCRs. So I thought it was time to look at this and see what is actually the contribution of individual parasite to the population. So to start, we used 4 of these clonal population that I generated in that first work. So 2 that were what we defined as single, so with a main VAR gene on, and 2 that we defined as low many with this like really low expression of multiple VAR genes. And then the first thing I did was DropSeq, which is a droplet-based technique, in principle similar to 10x, in the sense that they also, in this case, cells are encapsulated in a droplet, so individual cell with individual bead, and then you can generate the transcriptome of an individual cell. And I did DropSeq because we had already a setup in lab, in our neighboring lab that already did all the optimization. So it was really easy for me to first check with this technique. And I want to show you what we saw from the point of view of VAR genes. So again, starting with these 2 populations, if we look at the high single, and we look at, this is the transcriptome of VAR genes in an individual cell, you see that this cell, for example, has are off, except for this one that it's on. And this was actually true for several of the cells in this population. But then we saw also outliers. Let's say this one, where there was at least a second VAR gene on top of the main one, like this one as well. starting with the percentage of these three states was different. So you can see here that when you look at the low, many states, almost at least 75% of the parasite was falling into this low node category. And so obviously everyone in the field always thought that each individual parasite looked like this. So I was curious to look a bit more into these other two states that sort of seem to go against this principle of mutually exclusive expression. So I'm going to start with the multiple first. Initially, when we started seeing this parasite, we thought that it's possible that these are individual parasites that are in the process of switching from one virgin to another. And I started thinking about this question of like, how does really a parasite decide to which virgin to go next, to what should be the next virgin to turn on? And to explore this question, I took advantage of a mutant that we already had in lab, which is a knockdown of this enzyme called SAMS. And SAMS is the responsible for the production of SAM, whose in turn is the main methyl donor for histone methylation. So when we knock down SAMS, in turn, we reduce overall histone methylation in the parasites. And when I took this mutant and I looked at what happens at virgin expression, we start to see that at the population level, we start to observe several virgins turning on at the same time at a pretty high level. But this was not only true at the population level, it was also true when I did single cell on this, because you can see here, basically individual cells are really mirroring the same image as the population. What I think it's particularly interesting here is that I sort of expected that when you would lower histone methylation, all virgin would turn on at the same time. Whereas there is clearly some virgins that are more prone to be activated than others. And this is also interesting because it changes if I repeat the same transfection in a different clone. It's the same background, the same strain, but different clone. I see different virgin turning on, which sort of suggests that there is that not all virgins are created equal in these clones. And if that's the case, the question is what determines the propensity of activation of an individual virgin. And I just want to show you some preliminary data that we have on that, on a specific virgin that is called VAR2CSA. So VAR2CSA in general has been always a very interesting virgin because it's the gene responsible for encoding the PFMP1 who causes pregnancy-associated malaria. So a lot of work has been done on that, but I was interested in that from a different perspective. I was interested in that because we basically started observing that different clonal population, when we were growing them over time in lab, completely isogenic. So they are just daughter clones of the clone tree that I showed you before. They had a different propensity to activate this virgin. So you can see this clone 1 is very similarly expressing this green virgin, whereas clone 2 seems over time to go to VAR2CSA. And this is not something that we only observed. It's actually a fairly weird thing that in the field has been always discussed, like people that say, oh, all my parasites go to VAR2CSA over time. And other people that say, I've never seen that happen before. So it's really weird. So I was curious to see what could be causing this difference between these completely isogenic clones. And my first idea was because they are isogenic, maybe there is something different at the epigenetic level. So we took these different clones and we did the cut and run, which maybe you are familiar with, but is a similar technique as a principle to our ChIP-seq. So you basically permeabilize your cell, the antibody of interest binds to wherever it binds, and then through calcium activated MNase, you can basically cut the region of the DNA that are associated with your antibody of interest. And then the outcome is very similar to a ChIP-seq, so you can map where this antibody was binding within the genome. So I did that with H3K9 trimethylation. And this is just to show you how this metal mark actually looks in Plasmodium chromosome-wise, which is kind of weird compared to like other eukaryotes. Like in other eukaryotes, H3K9 trimethylation is like fairly, it's changing if a gene is silenced or not. So if a gene is silenced, it's normally it normally has this mark, and if it's active, it normally doesn't have. Whereas in Plasmodium, this mark is just devoted to clonally variant genes. So like var genes and family like that. So you can see that it's just basically in the subtelomeric region and a couple of clusters within the chromosome. Then if I zoom in to how that mark looks like at VAR2CSA locus. So here you can see that if the clone is actually expressing VAR2CSA, there is an overall reduction in the mark. But then between the two clones, and I did this when they were still expressing a different VAR gene than VAR2CSA, you basically see that there is almost no difference at the promoter region. But there was a clear difference here at the intronic region of VAR2CSA. And initially we were a bit puzzled about that, but I think it can make sense because if I show you the structure of a VAR gene, so a VAR gene is a biexonic structure. It has a main promoter that is the one that makes for the main mRNA when the VAR gene is on. And then it has a second promoter activity within the intron that makes for two non-coding RNAs. One that is in the antisense direction, and this is thought to be connected to the active VAR gene at the same stage where the active VAR gene is on. So let's say the first 24 hours approximately of the cell cycle. And then there is a second non-coding RNA that is also coming from the intron, but it's thought to be transcribed later in the cell cycle, which is when all the VAR genes are normally off. And again, another fairly conserved opinion in the field is that these non-coding RNAs comes from all the members of the family in the late stage of the parasites. But then when I started looking into that, the truth is that no one actually knows if they come from all the VAR genes at the same time, because this part of the gene is fairly conserved. So it's not that easy to design a probe that only targets one of them. So majority of the work was done with probes that would recognize several of these exon 2 at the same time. So I started to be curious because of what we observed with the cut and run, if it's actually true that all the VAR genes make this non-coding RNA equally. So I designed probes that were basically designed to be specific for individual non-coding RNAs. So in this case, this probe was specific for the VAR2CSA non-coding RNA. And what you can see by qPCR is that in the clone that wasn't activating VAR2CSA, there is clearly a higher abundance of this non-coding RNA compared to the clone that over time ended up activating VAR2CSA. And this was not only true by qPCR, but also when we did RNA-seq, we saw a clear difference between these two clones. Which, first of all, suggests that probably is not true that all the VAR genes make this non-coding RNA at the same time. I actually started also looking at other VAR genes, and I see that there are clear differences between some that make them and some that don't. But also that this could be potentially connected to the propensity of activation of a VAR gene. So if you think about this, the clone that had the less non-coding RNA seems to have a higher propensity to activate that certain VAR gene. So I think there's a lot to find out about this, but I want to just go through what I showed you so far. I think by looking at this by single cell level, we definitely saw that mutually exclusive expression rules are way less strict than we thought before. And that there seemed to be a different propensity of certain VAR genes to turn on, which might be connected to the expression of this exon 2 non-coding RNA. And I think this first part, it's really important for my first question. So like, how do actually parasites coordinate their VAR gene switching? So I think it's possible that the fact that there is a different propensity of activation is what actually guides to what VAR gene to go next. If you have some question about this first part, we can discuss now or in the end, whatever. Yeah.
Did your single cell and you see one dominant gene and then potentially multiple low expressing genes? Are you really sure that those aren't doublets or multiplets? Can you be sure that it's not?
So In the case, actually, I'm gonna go to the next slide just because it has that. So in this case, you mean, so in this case, we actually wondered that as well. We were very careful in growing the parasite in a way that there would be like almost no doublets, in the sense of doublets of two rings in the same cell. In the sense of two parasite in the same droplet, like the person that optimized this also did a lot of optimization in that sense to check that there wouldn't be that many doublets. But also what I think it's was weird is that when we were looking at doublets, there was doublets at this multiple, there was always the main vaginal along with another one, let's say. But then I never saw, let's say, this and another one together. Like it was always this one along with the second one, which I think if it was just doublets that we will say, we would then eventually see also one that has this one with another one, but that was almost never the case. But then I also, so in the case of the dropsic, I think the sensitivity was also not good enough, which I'm gonna go to that in a second. But we observed that even more if we were looking with different techniques. So like the percentage of double or at least multiple was too high to be just two cells in the same droplet. If it makes sense.
Second question. So I'm pretty convinced you're seeing multiple VAR gene expression in the same cell. Do you think you're getting two different proteins? Are there two different PMPM ones?
It's a good question. Maybe we go back to that after I show the second part, and we'll. In the case of the multiples, the right answer is that I don't know for a fact, in the sense that we would have to have antibodies that recognize different isoform of PFMP1, which we don't have. So I cannot guarantee that that's the case. But maybe we go back to this question afterwards, and we can tell you what I know at least.
So just two quick ones. So, I'm getting flu because you just said that, you can get the partial transcripts that are not doing much, the long non-coding kinase. That would presumably look exactly the same if you look at that on through prime end. Did you factor for that?
When
Did you
I
get any indication that those are two full transcripts? Of,
Yeah, I didn't, like we checked that
Honestly,
because,
no, that's-
yeah. I also in this case, I was also very careful at everything we mapped was uniquely mapped. So I basically discard every read that was going to multiple VAR genes, and that would often discard a lot of these exon 2 because they sort of very similar to each other. So majority of the reads we actually get from the first part of the VAR gene, which is fairly unique. But I also saw that we always check that the reads were sort of covering more than just the exon 2. And the other thing is that everything I did here was on ring stage, because that's, so basically VAR gene gets expressed in the first 24 hours, then they go down. Whereas these non-coding RNA seem to come up later in the cell cycle. So in theory, there shouldn't be that non-coding RNA expressed in the first part, where I did, they were tightly synchronized to 16 to 19 hours.
Thinking about your potential as called poised gene that is expressing and is sort of partially heterochromatized, would you just speculate, is it still being part of the heterochromatin body that is outside of, on the side of the nucleus? Or is it somewhere?
It's a good question. I feel like that's also something that I would be interested in looking more, that there are these ideas that VAR gene, similar, let's say, to trypanosome, are clustering in a spot in the nucleus. I don't know. I feel like it was looked a lot in the past. No one really revisited with the new techniques that we have to really look at how this VAR gene cluster. So, but potentially, I think it would be interesting to think that maybe this non-coding RNA might be also a way to either cluster VAR gene together or not, and that defines if in the next cell cycle that VAR gene gets activated or not. But this is pure speculation. I don't know that. Okay.
Can you clarify something before we move on, just to see that I understood? So each on VAR gene is the only one that's going to have this long non-coding transcript in the later stage, or all virgins actually have this long protein in each cell.
So this part, the long coding RNA, I didn't look at single cell yet, so I don't know that. The theory is that this one, it's only made from the active VAR gene. So it would be just activated in the first part of the cell cycle, just from the active VAR gene. But no one looked at this individual cell. This is all population based. And this, the one that I talked about, this would be made in the second part of the cell cycle. In theory, from all of them, again, this was what was taught before. Now I think that actually might not be the case. It might be expressed only from some of them. Now go on, then we can go back to these questions if you want. So with the second part, I would like to go a bit into this low null parasite, because I was particularly interested in them. Because obviously these are parasites this is a state that would be impossible to identify if you look at the population at the population level. Like this would always be masked by parasites that are actually expressing a VAR gene. But I was a bit scared that there was a limitation of like dropsic sensitivity. But I was kind of curious to see, if you think about what I told in the beginning about this, for example, the olfactory neurons, that they saw that in this stage where they had the really low level of olfactory receptor gene expression, they see this multiple low level expression of olfactory receptor genes. So I kind of was interested in seeing if we see something similar in this low null parasite, if we go with a technique with a better sensitivity. So in order to overcome that, I firstly, in collaboration with IDT, we designed enrichment probes. So there were probes that were just designed to enrich for VAR genes and some control genes. So I basically use these probes and apply them to the single cell DNA library that I prepared with Dropseq. And then I check after amplifying and sequencing how they look like. And this enrichment actually worked pretty well. And here you can see how that basically more than 90% of the transcript before enrichment were on genes that I didn't target with enrichment. Whereas after enrichment, up to 85 or 90% were either VAR genes or control genes. And this was also really specific because here you can see the 5-6 controls I used. And you can see that they were all enriched except for the CSP, which is a gene that is expressed at a different part of the life cycle. So it shouldn't be transcribed at this stage. And I didn't enrich it at all, despite having probes for it. And this allowed me to try to look at these cells in a similar way as the olfactory receptor people did. So this is very similar to what they did, just with VAR genes. So again, each column is a different cell, and each color in the column is a different VAR gene. And this is how they looked before enrichment, so just by Dropseq. And then after enrichment, this is what we got. First of all, you can clearly see then the enrichment worked, but also individual cells in the I single clearly had a major VAR gene on. But then we start to see that this background transcription of other VAR genes, which was even more obvious in the cells in the low man. So we start to see something similar to what they observe again for trypanosome for olfactory receptors, that this cell seem to have this low expression of several VAR genes. But when I made it to this point, I was, if I want to go on to check this, I cannot keep all the time I want to check a cell, I have to do Dropseq and then do enrichment. It becomes too expensive. So I was searching for basically an alternative method that would have a better sensitivity, that didn't require the use of enrichment probes, because as I told you before, these VAR genes are very different between different strains. So basically every time that I do a new strain, I would have to design a new set of probes, which is not feasible. And at the time I was starting to think about, I would like to check this also in field samples. So I was searching for something that would be a bit more portable and flexible. So I came across this device that is called a Hive. It's literally like a pocket lens device, very big like this. And it has individual picowells, where in each picowell there would be a bead. And then you can load the parasite on this device at a dilution that individual parasite goes in individual wells. And the cool thing about using this on the field, I think, is that once you load the parasite on the device, you can freeze the device and either process several of them like weeks after, or even ship it somewhere else for someone else to do the processing. So when I met someone that was using this, I thought, oh, this could be actually a good thing for the malaria field, where like field studies are not necessarily that straightforward. And so I decided to use this, and here you can see just quickly how better it worked compared to Dropsic. Like you can see that there was a nice increase in both cell recovered and UMI per cell. UMI are the number of transcripts per cell, despite loading basically a magnitude less of cells in the hive. And this obviously allowed us to do way more deep analysis on the transcriptome that we got that we couldn't really do after Dropsic. For example, we were able to actually put this parasite in UMAPs. So you probably are familiar with this, but the UMAP is basically a map where each dot is an individual cell, and the closer they are to each other, the more similar their transcriptome is. So when we got this out, the first thing we noticed is that this UMAP looks like a gorilla, which is something that someone told me, and then after that I can only see the gorilla. And that parasite actually cluster based on their VAR gene expression. So you can see here, this entire population I single A, it's all here in the back of the gorilla, and the head of the gorilla, it's all parasite from the high single B population, whereas the two low many seem to cluster a bit in the middle. And this entire clustering was basically driven by VAR genes, because you can see here the expression of a VAR gene. It's all the cells here are expressing the same VAR gene, and all the cells here are expressing the same, and even the little legs here are all defined by an individual VAR gene. Which was even more clear when I repeated this entire analysis by removing the VAR genes, you see that all the cells basically collapse into this blob, really meaning that there wasn't much of a difference between cells except their different VAR gene profile. And the better data we got from the hive allowed us to basically replicate what we already saw with the enrichment. So that individual cell really seemed to have different expression of different VAR genes in individual cell, but not at a particularly high level. So when we make it to this point, I was asking myself a very similar question to what Claire asked me, so what does this mean in terms of having a PFMP1 on the surface? So if the cell, let's say, that are in the single state, they look like this, do the other one actually still export a PFMP1 on the surface or not? And in order to investigate that, we took advantage of these broadly cross-reactive IgGs. They were isolated like 30 years ago from more than 800 adults in Malawi. And the idea here is that if you pull these IgGs from so many different adults, you should cross-react with a lot of isoform of PFMP1. So what we saw when we used these IgGs with the flow cytometry is that the parasite in this single state, they clearly react with the IgGs, whereas the parasite in the null state basically react the same way as an infected rapid cell. And this wasn't true for this example, but also when we looked at different clones that had different expression of VAR genes, and that there was a really nice correlation between the total VAR expression in blue and the amount of recognition by antibody in pink. Really suggesting that this null parasite seemed to have reduced antibody recognition. Which, if we put it together, I think on top of the things that I already told you before, I think this single cell really proved that parasite can exist in a null state, and as this null state seemed to render them less recognizable by antibodies. Which, if we go back to what I talked about in the beginning, and we think about this in the context of an infection, I think a lot of the work has been done, and also what I showed you in the first part, in understanding what happens here during an acute phase of an infection. How do parasites transition between different VAR genes? But not a lot is understood what happens in more chronic malaria infections. Because it is known again that asymptomatic falciparum infection can last up to a decade or longer. But also I think it's even more interesting, there are several examples of patients that were living in an endemic area and then left for several years. And then they got, for example, splenectomized, they got immunocompromised, they got pregnant. And you can see basically a relapse in malaria, in falciparum malaria in this case. So, which means that these parasites were able to survive undetected for an extended period of time, and they are still potentially, it can recrudescence once something happens, like you are immunocompromised. And I think that is my hypothesis is that these parasites that are able to do it, this might be these null parasites. Because I think if you think about this in the context of an infection, once our antibody have taken control of the different isoform of PFMP1, this null parasite might be the one that survive undetected for an extended period of time and maybe are able to come back. So this is what also I want to do next, just a couple of slides of what are my future directions. So I want to investigate this idea that these null parasites are the main drivers of these chronic malaria infections from two main points of view. One would be the how, so what are really the molecular mechanism behind VAR genes activation and silencing, bringing back to the London coding RNAs, as I told you before. I want to combine that with understanding the role of R loops in this, and what we sort of hinted to before also, I think with the novel techniques that we have to really look at chromatin organization, really look at how VAR genes are really with each other in the context of a nucleus. And then the other obvious question is, okay, if it's true that these null parasites are the driver of this chronic malaria infection, where are they? I think that possibilities are that these are circulating at a very low level. These asymptomatic individuals are like, they are basically undetected parasitemia. So if they are circulating, would be really, really low. So hard, but it would be nice to see, to compare samples from symptomatic and asymptomatic adults and see if we can identify these null parasites. But I think the actually more appealing ideas is that these null parasites might not be actually in circulation, but they might be collecting in extravascular reservoirs. So there is this, I think we have known for a long time that falciparum is in the bone marrow, and that's where they do gametogenesis. But there are also asexual stages in there, so no one really looked at these sexual stages to see what's their VAR gene expression. But then I think more recently, there has been a lot of studies that shown that there is a reservoir, a big reservoir within the spleen. So to me, conceptually, would make sense that this parasite, if they don't express a PFMP1, so they are less cytoadherent, they could accumulate within the spleen over time. And with this, I would just like to thank my lab and our two neighboring lab, and you all for your attention, and I'm happy to take other questions.
Questions, and for those online, as usual, just raise your hands and I'll unmute you, or you could put your questions in the chat.
Fascinating, really exciting. I love the gorilla. So two questions. So one was, I didn't understand how you did the clustering with the gorilla graphs. So if you are looking at parasite lineage, wouldn't there anyway only be different because of the PFMP1 and not, like, so if you had a whole population of parasites from the field and everywhere, it wouldn't actually be virgins that drive the clustering? I don't know.
I think it's a good point. The important thing is that a lot of people that do single cell, if you think about any single cell that you've seen in, like, I don't know if you've ever seen malaria parasite single cell, but they do this nice circle.
Yeah.
So the difference is that majority of the people load parasites that are like at different stages of the cell cycle. Whereas in this case, I was interested to see if I load this parasite that are really tightly synchronized, and is actually VAR genes the only thing that drives the difference. It makes sense, but I didn't know from the start. Like I was actually curious to see, okay, these parasites that are in this low many state, is there anything else that is actually different at the point of view of the transcriptome, but I didn't see any other difference than VAR genes.
Okay. And is there any known case of non-parasites in the field?
So not a
Or
lot,
multiple, sorry, or multiple.
not a lot of people have looked at, it's really hard to look at VAR genes in field samples because it's not, it was harder to do field studies in the first place, but to look at virgins, you not only have to take the sample, but you also have to build de novo the genome, because virgins are different for any strain that you look at. So there is not a lot of work that has been done on virgins at single cell. There, one example, which wasn't done at single cell though, was someone that got a case, one of these cases of like malaria after like multiple years that they were away. And someone took those parasites and put them in culture, and they saw that they were in this like low man state at the population level at least. But then when they were growing up for a little bit over time, then they started to expressing a virgin again. But that's it, literally the only case that someone looked at that, and it's like from like 15 years ago.
So is this a person that had the spleen removed?
Yes,
or-
exactly, yeah.
Okay, so then follow up, sorry, last question. In terms of, say, because you're culturing them for long term, there's no selection pressure. So if anyway, there was any sense of selection pressure, not necessarily PFMP1 antibodies, do you think that that then would really not exist? So in this your hypothesis of these very low chronic infections, there would still be some level of selection on antibodies, or maybe not because it's too low. So what's your thinking of the fact that you've selected them for long term without selection
It's-
pressure?
That's a very good point. The fact that what we see is just the culture of parasites. So I think it's a obvious next step would be, does this have any relevance for field samples? But I think what we saw with the IgGs made me think that these parasites seem to like not be recognized as well by antibodies. So even if there is an immune pressure, I think that because they are in red blood cells, so red blood cells are not really immunogenic by themselves. So the PFMP1 is really what makes them immunogenic. So maybe if they are really able to silence the PFMP1 on the surface, they wouldn't get recognized by immune pressure anyway.
Can I ask a follow-up question, Francesca, that probably exposes my ignorance about Plasmodium, but if the Plasmodium doesn't express a VAR gene, what do the knobs on the surface of the red blood cell look like without VAR? Because it's not just VAR and PFMP1 that's there, and how then, if there's still knobs, how are they not cleared by this?
It's a good question. We don't know that because I feel like up to, I don't know, 2 years ago, no one actually thought that parasite would exist without expressing a VAR gene. So I don't know. It would be interesting to look at this parasite and see if they still form knobs in absence of expression of
We don't
VAR.
even know if there's no knob, if they're completely back to
No,
normal.
no.
Okay.
Connected to both Priscilla and Richard's comments. I was just wondering, so maybe I missed it so are you able to clone out the null parasites and do they switch back? Do they express VAR genes in the next cycle or something, or have you been able to?
So I can't really, everything I did when I did the cloning, those are still populations. I can't right now take a population and just extract the ones that are purely null. It would be cool because I think that would be a pure null population, whereas now I just have the one that look null at a population level. But
Okay.
yeah, so I don't know that. My only example would be the one that I was talking with her before about the person that their spleen got removed. In that case, the parasite initially were silent and then
they
they
came back up.
reactivated the VAR gene. So it seemed that they can do that, but
These,
Example would observe in this specific Falciparum, or the other malaria parasite to be able to cause similar tricks when it comes to antigenic expression. Particularly, I'm thinking Vivax, if you know those sit.
so the thing is that Vivax has always been like it's always been less of a surprise, the fact that we're able to prolong their infections because of the hypnozoites.
Yeah.
So because in the cases where they look at the spleen samples, they see that actually was a paper that initially was mainly concentrated on Vivax, and they saw this big reservoir of Vivax in the spleen. So the fact that they are in the spleen seems to be unrelated to Vivax or Falciparum, but I think Falciparum needs to have a different trick compared to Vivax to prolong infections.
So I had a follow-up to the other discussion that you're having about how to isolate the VAR genes. So the knobs are essential for gelatin purification. So
so they wouldn't be captured by gelatin. And if you do that with some alkalis, gelatin doesn't work very well for purifying. So that might be a way
Mhm.
that you could find. And my second question was about these strange individuals that have been selectorized. So what's the advantage to a parasite starting to express the biology when there's no spleen?
I don't know, that's it, it's actually a good question, because, but even in culture, you would imagine that they don't have any reason to
No,
express a
it's actually-
gene in culture for a long time.
So we shouldn't be surprised if they stop expressing biology
Yeah.
in culture, because there's no point. But in the person with no spleen, why would they suddenly switch to turning the biology?
So they, in the person with no spleen, so they took the parasite out, and then in culture, they started re-expressing the gene. No.
That makes sense. Right, so makes sense.
I just have two questions out of curiosity. What do you think about the sensing within the patient, within a infected individual? The sensing of the microenvironment, the immune response around, like the spleen characteristics of the spleen in the brain. You thought about chronic infection, but let's say an acute infection, parasites in the spleen might be expressing different patterns, like would have different proportions, I would say, of this multiple, single and null parasites compared to the brain. My second question is, if you looked, we can talk about this later, just based on some preliminary data we have on parasites in the spleen. Did you look at Rossetti? Like, is known that parasites somehow VAC gene expression can affect the potential resetting of the infected cell?
So for the first question, I think it's a kind of a recent concept, the fact that how the environment can influence gene expression. Like for a long time, I think people just thought that gene was kind of random compared to like the environment, whereas I think now there are more and more data of people that change the, I don't know, the media where the parasite were grown, and they saw a response in terms of both making gametocytes, but also response in gene expression, which is fundamentally that SAMS knockdown that I showed you before was a paper that was looking at that. So if we mock with the media, do we see a change in expression? And we definitely know that there are certain triggers that seem to push for expression of R2CSA, which there is a part of Kirk's lab that is working on the connection between the placenta environment and the expression of R2CSA. So is there something that parasites sense in the placenta that triggers expression of R2CSA? But I think it's a very fresh field, so we don't know. I think it makes sense to me. It could also be that these parasites in the spleen are sensing, even the spleen itself is a very fresh field. So do we know if the parasites are sensing something in the spleen? Why do they accumulate there? And so I think they're a very good question that would be cool to answer. Can you remind me your second question? So the parasite that I did majority of the work, the first part, they don't rosette, and also that they don't even do knobs. So I can't really say if those one are rosetting. The second part I did with IT4, which that are still making knobs and should be able to rosette, but I didn't see anything in culture, but I don't know.
Surprised, Gilbert may have some thoughts on this, but surely not expressing the FBMP1, we see the purpose of the FBMP1 as being avoiding splenic clearance. That can't be the only reason for it. And if you're in the spleen, you're done, right? You can't transmit from the spleen. You've got to go somewhere else. So I'm presuming that part of the surface proteins is the environment, high CO2, low shear stress, but also like to enable you to transmit. That's something you're working on. Surface expression links with automation. Okay. Okay.
I think you make a good point. The fact that in theory it's not very advantageous to a parasite to accumulate that much in the spleen. Like there should be still parasites that eventually, if they want to keep transmitted, that they eventually go around. But maybe it's like it's a sort of a reservoir.
Sure,
Yeah.
it's great, like tactic to like, but it can't be your only tactic.
Yeah,
Yeah.
Any questions? Okay, there are no more questions. Thanks again, Francesca.
Thank you.
Before you guys leave, so just to remind you, there's two, this Friday, there's a rum. Mariana from Sheena Lab is going to present, and next week, Friday, another rum from, yet unannounced surprise, member from, Howick Lab, Sheena Howick.